Hello, welcome to the next A-level lesson from the conscientious biologist, Ben Gallagher. This starts our section on DNA, and this is DNA biochemistry. So we're going to look at nucleotides, which are the building blocks of DNA, and we're going to look at how they polymerise to form DNA strands and how that builds all the way along to chromosomes that you're familiar with inside your cell. As always, please do subscribe to my YouTube channel if you haven't done so already. Have a look at my Facebook and Instagram pages for extra biology knowledge that can round out your full information of the biological world. Thank you. So starting off looking at the structure of DNA, we're going to do a GCSE recap because quite often DNA isn't taught in massive detail at GCSE. And I always find as well, because it's quite little and delicate and there's lots of detail, students never tend to revise it very well, or certainly don't seem to retain the knowledge. So when they come up to me at A level, I always find I need to do a big GCSE recap anyway. So that's what we're going to do. So if we're looking at DNA, think of it as a code. It's like all written languages. But instead of letters, like we've got on the screen here, we've got INSTEAD for instead of letters, it's written in chemicals called nucleotides. Now, if you're struggling to get your head around that, remember a code can be made up of absolutely anything. Morse code is sounds, it's dots and dashes. Smoke signals, you can send out puffs of smoke of different sizes to convey messages to each other. Facial expressions are a type of code that you can read to understand how someone is feeling. Codes are anything that convey information. DNA, it's written in chemicals called nucleotides. Now, there's four main types of nucleotides. So unlike the 26 letters we have in our alphabet, you've only got 4 with DNA. Now at GCSE, you should have covered at least the letters that they start with, A, G, C, and T. But the chemical names, which you must know at A level, are adenine, thymine, cytosine, and guanine. You must know those. Now, if we're going to start to form those into a genetic code, it could be something like this, that GCT, CAGG, TAG, CT might mean something in genetic language, that code. And if you think back to the GCSE lessons and what we mentioned in the first cell biology lessons in the A-level ones, DNA codes for sequences of amino acids, which will fold up into the correct shape to become functional proteins. So what we're talking about here with this strand, with this piece of genetic code, is that this one would code for 4 amino acids because each amino acid has a 3 base name. So GCT at the start there would be a code for an amino acid. The 3 base name at A level is called a codon, or that's just what it's actually called, a codon. We'll get to that when we look at protein synthesis in more detail. Okay, but that's a piece of genetic code because it codes for a sequence of amino acids. Now, DNA is double-stranded. Again, you should know this from GCSE, that it's double-stranded because the second strand forms by something called complementary base pairing. If you look at the shape of the nucleotides that I put over there, you can see how they're in pairs and they match up. That A is complementary in shape to T. You can see one's kind of a V-shaped point, one's a V-shaped going in as a receptor for it. C is complementary to G. Again, the shapes match. So if we're going to build a complementary strand onto this one, you should be able to do this yourself. You can see the C's and G's will match up, the A's and T's will match up, and we get this double strand of DNA. Now the double strands of DNA will twist themselves into this structure, which is a double helix, which should be more recognizable to you. The double helix can then curl itself up even more and coil and coil and coil until you've got chromosome, and chromosomes are found in the nucleus inside a cell. Now, everything that's on the screen now, apart from possibly the actual names of the 4 bases over there, the chemical bases on the nucleotides, I would expect you to know everything that's on this screen. If it doesn't look familiar, watch this again or go back to my GCSE video, which explains this slower and in more detail and in more step-by-step. But make sure you know this for carrying on. Now, this is a GCSE recap. We've already included a few extra details with the names, but we're going to put a few new terms on as well, a few extra ones. So looking at the DNA strand over there, we can label each strand to a coding strand and a template strand. Now that becomes relevant when we start looking at how DNA is used. We'll start to talk about that in the next video on DNA replication. But we're going to add some labels to this as well. What you'll see here is that the double helix starts to wrap itself around those kind of purple blobs. Those tiny little purple blobs there are histone proteins. We mentioned those in the cell ultra, or in the prokaryotic cell ultra structure video, because we said how eukaryotic DNA wraps itself around histone proteins, these, but prokaryotic DNA doesn't. It's naked DNA. It doesn't wrap around these histone proteins. Now, if you look really carefully at this diagram, those individual little purple blobs are histone proteins. But look at what's happening. The histone proteins form little clusters of 8. And the cluster of 8, as it shrinks in size, look, the cluster of 8 histone proteins is called a nucleosome. Again, another term that you need to know. And once you've got DNA wrapped around histone proteins, you refer to it as chromatin. Okay, so DNA double helix, the DNA molecule wrapped around histone proteins, is called chromatin. Chromatin then further wraps itself up to form a chromosome. So this page now has GCSE recap detail with a few extra A-level terms. So take a screenshot of this because it's really essential. You know absolutely every detail that's on this screen at present. Right, let's start to get properly into the A-level detail now by looking at nucleotide biochemistry. So those shapes, they're on the far side of the previous scheme, the yellow, green, orange and red shapes. Those are nucleotides with the base sticking out that have that specific shape. But let's look at what those molecules actually look like. So initially they've got this bit in the center. This is deoxyribose pentose sugar. A pentose sugar is any sugar that contains 5 carbons, like you can see here, okay? But this one is specifically called deoxyribose. It's the D from the name DNA. So you can see I put in red the DNA, but it's the deoxyribo. That's the sugar that's in it. And then it's a nucleic acid because it's in the nucleus and it's also slightly acidic. So DNA stands for deoxyribonucleic acid, the deoxyribo coming from deoxyribose. That's the sugar in the middle of it there. Now, unlike the triglycerides that we looked at in previous videos on membranes, you don't need the full biochemical detail of this, but there's a few bits that I want you to know. Firstly, that top point has an oxygen on it. That is quite important that I want you to know. Now, we mentioned that a pentose sugar is a pentose because it contains 5 carbons. So let's put those carbons in place, circling around on there. So you'll see there's a carbon on each point and a carbon on the top of that bit that sticks out. For a very important reason that will become clear on one of the next slides, I'm going to number those carbons, and we number them in a clockwise direction starting after the oxygen. So you've got the C1 nearest to me, 2, 3, 4 and 5 over there at the top. Now, really importantly, look over at the far side, look at the top of it, it's the 5th carbon. On the bottom of the far side, it's the 3rd carbon, okay? 5 at the top, 3 at the bottom. 5 to 3, 5 to 3, there's a reason why I'm doing this, which will become clear. But 5 at the top, 3 at the bottom. Remember that, okay? I'm now going to get rid of those carbons because you don't really, for A-level, need to know they're there. But remember that 5 to 3, 5 at the top, 3 at the bottom. It's important, okay? I'm going to get rid of those and we're going to put the next component. So attached at the top over there is this big blob. That's the phosphate group. Exactly the same structure that you got in the phospholipid. So you've got a phosphate group up there. Again, you don't need to know it, just like you didn't need to know it for the phospholipid. We're just going to put it as a green blob. Coming out the side here, closest to me, which you may remember was the 1st carbon, we've got this. Well, that yellow shape should make sense because you saw it on the previous diagrams. That yellow thing sticking out is the nitrogenous base. Now, just to link that to the previous diagrams, look, that was what I was using to symbolize a nucleic acid, a nucleotide in my GCSE presentation, just because all you really need to know is that nitrogenous base sticking out. But I'm going to get rid of that again so you've got the proper structure. And remember, those nitrogenous bases can be adenine, thymine, cytosine, and guanine. So what is on the screen right now is what you absolutely 100% must be able to draw from memory and label all those parts, because you could be asked to do that in an A-level paper. Take a screenshot of this to add it to your notes. Okay, let's start to build that up now into a DNA molecule. So I'm going to pull this one up here, and you'll see I've added an A to that because my yellow shape in the previous slides, the A was an adenine. So this is an adenine nucleotide now. Now, if that's the start of my code, that's the first letter, the first chemical of my code. If I want the next one to be a guanine nucleotide, I've got to join those two together. And the join goes from the third carbon, so the bottom corner of the nucleotide, to the phosphate group. So I've just put that purple line, I've marked in the different color so it's really, really obvious. That is called a phosphodiester covalent bond. So the type of bond is covalent, its specific name is phosphodiester. I really want you to always label it as phosphodiester covalent bond, because some mark schemes want phosphodiester, some say you have to have stated covalent. If you write phosphodiester covalent bond, you're always going to hit the mark. Okay, so that's going to join those together. If I want my next nucleotide to be a cytosine, again, I need another phosphodiester covalent bond to join that together. Okay, that could just continue on down, making my coding strand. Okay, now remember what I said to you previously about the fifth carbon and the third carbon. DNA has a direction. You'll see I've put a 5, the little apostrophe that's called a prime. So that's 5 prime up at the top over there where that fifth carbon was. So down the bottom of the strand here, I can put in a 3 for 3 prime. Now DNA is read from 5 prime to 3 prime. It has a direction. Just like if you're reading a book in English, we read the sentence from left to right, left and right if things are just floating around. So DNA is always read from 5 prime to 3 prime. That's because of the carbons. Look, the carbon, upmost carbon was the fifth carbon. The bottommost carbon is the third carbon, 5 prime to 3 prime. So it's read in that direction. Now remember now, we've got to put on the complementary strand next to it. So I'm going to start with the one down the bottom here, starting at the bottom for a good reason. But you'll see it's a guanine to join to the cytosine because they fit, they're the complementary base pair. The next one is going to be a cytosine to join to that, and I've got to bond those together. Look at my nucleotides though, they're upside down. Now, there's a reason for this. It's something quite important for another part of biology to talk about. But if we put the top one on just for completeness, that's a thymine to bond to the adenine. Again, we'll put our phosphodiester covalent bond on. But look, they're the other direction now. It is anti-parallel. The 5 prime here is on the bottom with the 3 prime at the top. They don't run parallel, they run anti-parallel to each other. Okay, so the DNA double strand, whilst it would obviously be massively longer than that, it's not just going to be 3 long, it would be thousands long. But that's your basic structure, where you've got 2 strands anti-parallel to each other, joined by complementary base pairing. And the bonds that hold those complementary base pairs together are hydrogen bonds. Hydrogen bonds are quite weak bonds, which is good, because to read the DNA, just like reading a book, you need to be able to open it. So those complementary base pairings, those hydrogen bonds need to be breakable because you need to open the DNA for the cell to be able to read it. So that's really, really important. So lots of information on that diagram, more key terms, you've got to know all of that. If we extend that into a long strand then, so something a little bit more representative, still ridiculously short, but you can see how it's just that repeat structure all the way down that we've got on one side, the side in this instance further away from me, 5 prime to 3 prime coding strand, the anti-parallel strand going 5 prime to 3 prime at the top, that's the template strand. Take a screenshot of this so you understand exactly how the DNA strand forms, but then we're going to do a slide to put it all together. So really quickly then, let's link that last slide to the GCSE recap slide so we've got everything in one place. Here's the strand that we talked about in the last lesson. If we're going to add the labels to this, we've got the coding strand here, we've got the template strand. Bear in mind, the DNA strand inside a cell floating about can flip, can turn, so you can't always tell which is the coding strand and which is the template strand. You'd need to look at what it's doing to see the behavior of the strands to be able to label them accurately. Generally, in the exams though, they show the coding strand as going 5 prime to 3 prime left to right. So that might give it away in the exam. Whichever one goes 5 prime to 3 prime left to right, how we would read, that's probably going to be the coding strand rather than the template strand. If I put a little box over there around one nucleotide, you might be asked in the exam to label one nucleotide by drawing a box around it. I can also label a phosphodiester covalent bond. Of course, there's loads of them, but I'm picking that one to label. Within another little yellow box over there, you can see that I'm marking complementary base pairs. You could add to that on your label that they're held together by hydrogen bonds. If you're printing this out and you're writing on it, you might also want to label the deoxyribose, the phosphate group, adenine, guanine, cytosine, thymine, add as much detail as you want to. But linking this to the initial diagram, which if I pull back up, you can see how our double strand of correctly drawn nucleotides fits perfectly in at the start of the diagram here, showing then how it coils, how you've got the double helix, wrapping around histone proteins to form nucleosomes, which forms the chromatin, which coils into chromosomes, which are what you find in the nucleus of a cell. So here's a slide with pretty much all the detail on it. Print it out, add more detail to it if you wish. From here, go on to the second presentation on DNA, which looks at DNA replication. So that's about what DNA has to do prior to dividing by mitosis, because if you've got one cell and that one cell is going to grow and split into two, each new cell will need a full set of DNA. So at some point during the cell's life, it must double all its DNA. This is by a process called semi-conservative replication, and we'll look at that in the next video. If you've enjoyed this video, if you find it useful, please do give it a thumbs up, subscribe to my YouTube channel if you haven't done so already, do have a look at my Instagram and Facebook videos, and if you've got any comments or feedback on this, do think about chucking them in the comments. Thank you.